Good afternoon, students. In the last lecture, we have discussed about the layout of field experiment, okay, which is also called as the experimental design, okay, and also we have discussed regarding the two basic types of the experimental designs. The first one we have discussed the Latin square design, and another one is the randomized block design. In today's lecture, we are going to discuss about the biometrical techniques in the plant breeding. Today's topic for the discussion is the biometrical techniques in the plant breeding. Okay, and this is the last article in the unit first plant breeding. After which discuss the seed technology in the next lecture. Okay, this is the last topic in the unit first plant breeding, and in the next lecture we will start the unit second, and that is dedicated to the seed technology. Okay, today's topic for the discussion is the biometrical techniques in the plant breeding. Biometrical techniques in the plant breeding. Okay, the first of all, what is mean by the biometry? Okay, before discussing regarding the biometry, all you should know about the statistics. Okay, statistics, it is the branch of applied mathematics which deals with the collection, presentation, analysis, and interpretation of the numerical data. Okay, before going to discuss about the biometrical techniques in the plant breeding, you should know regarding the statistics. Statistics, it is the branch of the applied mathematics which deals with the collection, presentation, analysis, and the interpretation of the numerical data. While the biometry, which is also referred as the biometrics, it is the science that deals with the application of these statistical procedures to the study of biological problems. Okay, biometry or the biometrics, this is the branch which is also called as the biostatistics. Biostatistics, it is the science that deals with the application of the statistical procedures to the study of the biological problems. Okay, whatever are the statistical procedures, that is, these are applied to study the biological problems, and that branch it is called as the biometry, or it is also referred as the biostatistics. Okay, here in this topic, the we are going to discuss the brief description of some common biometrical techniques and their relevance to the plant breeding. Okay, because the biometry, it is a very vast topic. Various books are also available in the market uh, on the biostatics, but in this topic we are going to discuss. The present discussion, it is limited in scope to the brief description of some of the common biometrical techniques and their relevance to the plant breeding. Okay, generally the techniques which are used in the biostatistics, these are divided into the two parts. The first one, the experimental designs, and the second one is the statistical method. Okay, already we have discussed the one part, experimental designs. That is also the one of the biometrical technique which is extensively used in the plant breeding. Okay, the statistical techniques which are used in the plant breeding, these can be divided into the two parts. The first one is the experimental designs, or which is commonly called as the layout of field experiment, and the second one is the statistical methods. Okay, of which the first part, experimental designs, we have already discussed in the last lecture, and in today's lecture we are going to discuss the other statistical methods which are used in the plant breeding. Okay, statistics, it is the branch of applied mathematics which deals with the collection, presentation, analysis, and the interpretation of the numerical data. While the biometry or the biostatistics, it is the science that deals with the application of these statistical procedures to the study of the biological problems. Then what is the importance of the biometry or the biostatistics in the plant breeding, or how it is used in the plant breeding? Okay, the biometry or the biostatistics, it is the important branch which is used in every life science, in every experiment which is done in the life sciences. Okay, that's why it is very important. But here we are going to discuss how actually the biometry or the biostatistics it is used in the plant breeding. Okay, how it is used. Okay, the first of all, the biostatistics, it is used in the assessment of the genetic variability present in the population. Okay, biometrical techniques, these are used in the assessment of genetic variability present in the population. Okay, here in the bracket you can see the different measures of the genetic variability here. These are used, that is, by using this method the genetic variability in the population can be assessed. For example, here the range, then variance, standard deviation, coefficient of variation, and the standard error. Okay, these are the methods which are used to assess the genetic variability present in the population. Then second one, it is useful in the selection of elite genotypes. Okay, elite genotypes from the mixed population. Elite genotypes, or simply we can say the superior plants. Okay, elite. Okay, the plants which are vigorous and which are more productive. Okay, the biometrical techniques, these are used in the selection of elite in the selection of elite genotypes from the mixed population. Okay, here these are the methods which are used for the selection. These are correlation, path, and discrimination function analysis. Okay, these are the statistical procedures. Okay, and these are used in the selection of elite genotypes from the mixed population. Then also these are techniques, these are used in the choice of parents and also the breeding procedures. Okay, these methods, these are used in the choice of parents and the breeding procedures. These are the statistical procedures which are used, that is diallele, partial diallele, then generation means, triallele, quadriallele, biparental cross, triple test cross analysis. Okay, by using these methods the parents are selected, and then these are used in the breeding. Okay, then also these techniques, statistical techniques or the statistical procedures, these are used in determining the varietal adaptation. Okay, that is the for example, it is done by using the stability analysis. Okay, in determining the varietal adaptation, which is done by using the stability analysis. Okay, you may think what is the importance of the statistics in the plant breeding. Okay, in the plant breeding, that is, numerous observations are there, numerous numerical data is available. But this numerical data should be properly presented, it should be properly interpreted, so it will be useful to the plant breeder. And it is done by using the different statistical procedures. Okay, and the branch which is helpful for this one, this branch, it is called as the biometrics or the biostatistics. Okay, the numerical data which is available, it should be processed easily, it should be presented, it should be interpreted well, and it is done by using the different statistical procedures. Okay, there was biostatistics, it is useful in the assessment of genetic variability present in the population. It is useful in the selection of elite genotypes from the mixed population. It is also used in the choice of the parents and the breeding procedures, and also it is used in determining the varietal adaptations. Okay, then here, the first of all, the assessment of variability. Okay, what is meant by the assessment of variability? We are discussing here how actually the variation in the mixed population it is assessed. Okay, because the genetic variation, it is the the variation in the population, it is the important prerequisite for the any crop improvement program. If there is a if the variation is not present in the population, then it is not possible to improve the crop and to release the new improved variety. And that's why the variation, it is the most important prerequisite required for the crop improvement program. And that's why here we are discussing here the assessment of variability. Okay, how the variability in the plant it is assessed. Okay, here all you know that the magnitude of variability present in the crop species provides the basis for effective selection. Okay, just I have told you that it is the important prerequisite for the crop improvement program. Okay, the magnitude of variability present in the crop species provides the basis for the effective selection. Okay, here the phenotypic variation present in the population arises due to the genotypic and the environmental effect. Okay, that is generally the selection it is carried out depending upon the external characters, which are also referred as the phenotypic characters. And these external characters or the phenotypic characters, these are due to the mainly two effects. The first one, there is a genetic genotypic component, that is these character may be the due to the genetic constitution, or sometimes these external characters or the phenotypic characters, these are also influenced by the environmental effects. And that's why here the phenotypic variation it is referred as the total variation, because these variations may be due to the genotypic and the environmental effects. Okay, and thus its magnitude, the magnitude of phenotypic variation differs under the different environmental conditions. Okay, the magnitude of the phenotypic variation differs under the different environmental conditions. Okay, but here the genotypic variation it is due to the genotypic differences among the individuals within the population, and it is the main concern of the plant breeder. Okay, the plant breeders, that is these are mainly concerned with the genotypic variations. Okay, because these variations are heritable, that is these are transferred from the one generation to the another generation. But the variation, the phenotypic variations which are arised due to the environmental effect, that is they may vary due to the changing environmental conditions, and these are non-heritable, that is these cannot be passed, these cannot be transferred from one generation to the another generation. And therefore the plant breeders are mainly concerned with the genotypic variation. Okay, here the genetic variance, the genetic variance it refers to the heritable portion of the total variance or the phenotypic variance. Okay, total variance, phenotypic variance is referred as the total variance because the phenotypic variance it is due to the two effects, it may be due to the genotype or it may be due to the environmental condition. Okay, the genetic variance it refers to the heritable portion of the total variance or the phenotypic variance. Okay, then the genetic variance has been classified by the different scientists in the various ways. Here you can see the Fisher in the 1988 classified the genetic variance into the three types. The first one is the additive variance, second one is the dominance variance, and the third one it is the epistatic variance. Okay, while right in the 1935 classified the genetic variance into additive and the non-additive variance. Okay, here the additive variance, you can see here the additive variance and the non-additive variance, the dominance and the epistatic variance, these are here referred as the non-additive variance. Okay, right in the 1935 classified the genetic variance into the two groups. The first one is the additive variance and the second one is the non-additive variance. Non-additive, it is dominance and the epistatic variance. Okay, then Mather in the 1949 classified the genetic variance into the two types, that is the heritable fixable and the another one the heritable non-fixable. Okay, heritable, fixable, and non-fixable. Heritable, non-fixable. Here the additive variance, it is the heritable and fixable, while the remaining two, dominance variance and the epistatic variance, that is these are heritable and these are non-fixable. Okay, this is the classification which was given by the first of all Fisher in the 1918, Wright in the 1935, and the Mather in the 1949. Okay, we are discussing here the genetic variance, and the genetic variance, it is the heritable portion of the total variance, heritable portion of the total variance, or the phenotypic variance. Okay, here we are discussing the type of the genetic variance. Here you can see these are three types of the variance. The first of all, the additive variance. What is meant by additive variance? Additive variance, it is refers to the portion of the genetic variance which results due to the average effects of genes on all segregating loci. Okay, additive variance, it refers to the portion of genetic variance which results due to the average effect of genes on all segregating loci. Okay, then second one, the dominance variance. Dominance variance refers to the deviation from the additive gene action resulting from intra-allelic interaction. Okay, that is the interaction between the alleles of the same gene or the same locus. Okay, the dominance variance, it is the variance which is mainly arised to the interaction between the alleles of the same gene or the same locus. Okay, that is the dominance variance. And the third one, here you can see the epistatic variance. Epistatic variance, it refers to the deviation as a consequence of interallelic interaction. That is the interaction between the alleles of the two or more different genes or the loci. Okay, here that is the difference between the dominance and the epistatic. In the dominance variance, it is arised due to the interaction between the alleles of the same gene or the same locus. And epistatic variance, it is the variance which is arised due to the interaction between the alleles of the two or more different genes or the loci. Okay, that is all about the additive variance, dominance variance, and the epistatic variance. Here the epistatic variance, it is again of three types. Okay, epistatic variance is again of three types. Here it is the short form, here additive dominance, and the dominance type of epistatic variance. Okay, here the epistatic variance, it is again classified into the three types. The first one, additive variance. It refers to the interaction between the two or more loci, each exhibiting lack of dominance individually. Okay, it refers to the interaction between the two or more loci, each exhibiting the lack of dominance individually. Okay, here in the short form, it is denoted as a AA. Okay, that is the additive into additive, and it is fixable. It comes under the additive gene action. Okay, it is fixable and it comes under the additive gene action. Then additive into dominance epistatic variance. It refers to the interaction between the two or more loci. Okay, it refers to the interaction between the two or more loci, one exhibiting the lack of dominance and the other dominance individually. Okay, here we have discussed the additive into additive. It is here, it is the interaction between two or more loci, each exhibiting the lack of dominance individually. Okay, but here into additive into dominance, it is referring to the interaction between the two or more loci, one exhibiting the lack of dominance and the other is dominance individually. Okay, here both these loci, two or more loci, that is they exhibiting the lack of dominance. But in the additive into dominance, here the one it is exhibiting the lack of dominance, and the other one it is a dominance individually. Okay, that is the difference between the additive into additive variance and additive into dominance variance. Okay, here it is denoted as the additive into the dominance variance, and it is non-fixable. Okay, here additive into additive epistatic variance it is fixable, but here the additive into dominance variance it is non-fixable. Okay, then the third one, the dominance into dominance variance. Okay, dominance into the dominance variance, here you can see it refers to the interaction of the two or more loci, each exhibiting dominance individually. Okay, that means two interaction of two or more loci takes place, and each of the exhibiting the dominance individually. Okay, it is represented as the d into d, and it is also non-fixable. Okay, it is also non-fixable. Okay, in the natural plant breeding population, the epistatic variance has the lowest magnitude. Okay, that is another important thing regarding the epistatic variance, is that in the natural plant breeding population, the epistatic variance has the lowest magnitude. Okay, that is about the that is about the assessment of variability. Okay, we have discussed the different types of the variance in the populations. Okay, now we are moving towards the simple measures of variability, how the variability among the population in the plant breeding it is assessed or it is measured. Okay, there are different measures which are used for the assessment of variability, and these commonly include: the first one is the range, second one is the arithmetic mean, the third one is the variance, the fourth one is the standard deviation, fifth one is the standard error, and the sixth one is the coefficient of variation. Okay, these are the six measures which are very commonly used as a measure of variability. Okay, the first one range, second one is the arithmetic mean, third one is the variance, fourth one is the standard deviation. Standard deviation here it is referred as the SD, then standard error it is referred as the SE, and the sixth one is the coefficient of variation, which is also referred as the CV. Okay, here the first of all, first the simple measure, that is the range. Okay, very simple. Range it is nothing but the difference between the lowest and the highest values present in the observation in the sample. Range is nothing but the difference between the lowest and the highest values present in the observation in the sample. Okay, for example, suppose that the data is provided regarding the oil content of the 20 cotton seeds. Okay, suppose that the data is numerical data, it is provided of the oil on the oil content of the 20 cotton seeds. Okay, suppose that the highest value is 25% and the lowest value it is 50%, then the range will be 15% to 25%. Okay, that is the range of that particular numerical data. Okay, range it is the difference between the lowest and the highest value present in the observation in the sample. Okay, it is the simplest possible measure of variability, and its computation is very easy. Just by observing the data you can decide the highest value and the lowest value. Okay, and that is the range of that particular data. Okay, that's why here we are saying that it is the simplest possible measure of variability, and it is the computation is very easy. Its computation is very easy. You can easily compute, you can easily calculate the higher and the lowest value of the sample. Okay, but however it is a very crude measure of variability. Okay, though it is very simple, it can be computed very easily, but it is a very crude measure of variability, because it cannot consider the all the values it cannot taken into the consideration the all the values of that particular sample, and that's why the range, it cannot be used for the further statistical procedures, for further statistical analysis. Okay, that's why the range, that it cannot be used for the statistical analysis, as it is a very crude measure of variability. Okay, also the range not indicate as how the data behave in between the highest and the lowest value. Okay, it does not indicate how the data behave in between the highest and the lowest value. Okay, and that is the drawback of the range, and that's why the range, the value of the range, it cannot be used for the further statistical analysis, for the further statistical procedures. Okay, that is about the range. Then the arithmetic mean. Okay, or you can calculate the average, just that is, this is the arithmetic mean. Arithmetic mean, it is also known as simply as the mean or the average. Okay, here the spelling mistake is there. Please read as a mean. Okay, it is also simply known as the mean or the average. Okay, read here as a mean. It is also known simply as the mean and the average. It is very simple to calculate the average or the mean from the given data. Okay, just all you know, it is computed by dividing the sum of all observations in a sample by their number. Okay, here you can see the formula for the calculation of arithmetic mean. Here x dash, it is the sign which is used for the arithmetic mean, is equal to the, in bracket, summation x. Summation x means that is summation of all the observations, okay, divided. Okay, here n means the number of observations. Suppose that there are 20 number of observations, then the value of n will be the 20. Okay, it is calculated by the simple formula, arithmetic mean is equal to the sum of all observations divided by the number of observations. Okay, and as it all the values, these are used in this formula, all the values provided from a sample, that is, these are used in this formula, it gives the accurate central value, and therefore the arithmetic mean, it is widely used in the further statistical analysis. Okay, range is not used because all the values are not taken into the consideration, but in the arithmetic mean, as it is it is considering all the values provided, that's why it gives the accurate central value, and this is widely used for the further statistical analysis or for further statistical procedures. Okay, then here the third one, variance. Okay, variance, here it is defined as the average of the squared deviations from the mean, or it is the square of the standard deviation. Okay, variance is defined as the average of the square deviation from the mean, or it is the square of standard deviation. Okay, generally the variance, it is expressed as the sum of squares of deviations of all observations. Okay, here you can see the sum of squares of the deviations, it is the square of the deviations. Okay, sum of the squares of the deviation of all observations of a sample from its mean, and divided by the degree of freedom, that is the n minus 1. Okay, where n means the number of observation. Okay, this is the formula by which the variance is calculated. Okay, variance, it is denoted as the s square air. Okay, here you can see the summation x square. Here x means the observation. Okay, the summation of x square, the first of all you have to square the value of that observation, and the summation of all x squares minus, here you can see the summation x. Okay, into the bracket, it square divided by number of observations, and all these, it is divided by the degree of freedom n minus 1. Here it is the sign of summation. Here x means it is the observation. Here x square, it is the square of the observation, and the n, it is denoting the number of observations respectively. Okay, the variance, it is an effective measure of variability which permits the partition of variation into the various components. Okay, variance, it is an effective measure of variability which permits the partition of variation into the various components. Simply you can remember variance, it is nothing but the square of standard deviation. Okay, square of standard deviation. Here you can see what is meant by the standard deviation, the fourth measure of variability. Okay, standard deviation. How to calculate the standard deviation? You can just remember standard deviation. It is the square root of the variance. Okay, it is the square root of the variance. Here you can see in this formula, one standard deviation is equal to the under square root variance. Okay, under square root is the variance. It is the square root of the arithmetic mean of the squares of the deviations measured from the mean. Okay, in other word we can say the standard deviation, it is the square root of the variance. Here you can see the, in other word we are saying that the standard deviation, it is the square root of the variance. It is the best measure of variation in the population. Standard deviation, it is believed to be the best measure of variation in the population. Okay, then the standard deviation, it is based on all the observations of the sample. Okay, all the observations of the sample are taken into the consideration for the calculation of the standard deviation, and that's why the standard deviation, that is the measure which is used for the further statistical analysis. Okay, standard deviation, it is based on all the observations of the sample, and it is used for the further statistical analysis. Here the standard error. Okay, standard error, it is the measure of uncontrolled variation present in the sample. Okay, standard error, it is the measure of uncontrolled variation present in a sample. Okay, then standard error, it is estimated by dividing the estimates of standard deviation by the square root of number of observations in the sample. Okay, here you can see the standard deviation, it is denoted by capital SE, and it is equal to the standard deviation divided by the square root, in square root, number of observations. Okay, it is estimated by dividing the estimates of standard deviation by the square root of number of observations in the sample, and it is denoted by SE. Okay, here you can see SE means standard deviation, SD means standard, SE means standard error, SD means standard deviation, and here N means the number of observation. That is how easily the standard error can be calculated, and it is the measure of uncontrolled variation present in the sample. Okay, here the last one is the coefficient of variation. How exactly the coefficient of variation is calculated? Okay, the standard deviation is an absolute measure of variation, and it is expressed in terms of the unit of the variable. Okay, simply we can say the value of standard deviation, it is having the unit. Okay, for example, suppose that the data is provided, the observations in the data, these are provided in kilogram. Okay, then the value of the standard deviation will be in the kilogram. Okay, if the data or the observations in the sample, these are provided in the liter, then the standard deviation, the value of the standard deviation will be also in the liter. Okay, whatever may be the unit of that particular observation in the sample, the unit of standard deviation is same. Okay, the standard deviation is an absolute measure of variation, and it is expressed in terms of the unit of the variable. Okay, but For the purpose of comparative studies, a relative measure of dispersion or the variation is required. Okay, that means the relative measure, that means which is not having any unit. Okay, that means the standard deviation, it is having the unit of that variables. But here, this measure coefficient of variation, it is not expressed in any unit. Okay, it is never expressed in any unit. Okay, for the purpose of comparative studies, suppose that there are two samples are available. Okay, and we have to compare these two samples, then we will need a relative measure to compare the variation in between these two samples, and it can be done very easily by just calculating the coefficient of variation of these two samples. Okay, because the coefficient of variation, it is not having the any unit, but standard deviation, it is having the unit. Okay, and that's why for the purpose of comparative studies, a relative measure of dispersion or the variation is required. Coefficient of variation serves this purpose as it does not have any unit. Okay, coefficient of variation serves this purpose as is does not have any unit. The how it is calculated, the ratio of standard deviation of sample to its mean expressed in percentage is called as the coefficient of variation. Okay, the ratio of standard deviation of a sample to its mean, it is expressed in percentage, is called as the coefficient of variation. Okay, simply you can see how the exactly the coefficient of variation is calculated. Okay, coefficient of variation, CV, is equal to the, in bracket, standard deviation divided by mean into the 100. Okay, that is the simple formula to calculate the coefficient of variation. Okay, coefficient of variation, CV, is equal to the standard into the bracket standard deviation divided by mean into the 100. Okay, this measure, it is very useful for the study of variation in more than one sample or series. Okay, that is the coefficient of variation, it is very useful for the study of variation in more than one sample or the series. Okay, whenever the coefficient of variation is high, suppose that there are two samples, in one sample the coefficient of variation is high, then that we can say the observation, the sample is less consistent, or the these are having more variable. Okay, variation is more in that particular sample, and suppose that the another sample which is having the lower value for the coefficient of variation, then we can say the sample is more consistent and it is less variable. Okay, that is why how it is how the value of coefficient of variation, it is used to compare the two samples. Okay, when the coefficient of variation is high, the sample, it is less consistent or more variable, and when the value of coefficient of variation is very low, then the sample is more consistent or the less variable.